All right, our next step, the embryonic stage. So from zygote to embryo, so from week 2 to week 8, 2 months in, here's where we start seeing those life support structures. So this starts at implantation, so we need all of those resources that are going to help us survive. So the placenta and umbilical cord allow this embryo to start pulling resources from the mother's bloodstream. So it has sort of a protective function, so this can help if the baby has, say, a different blood type from the mother. This is supposed to help mitigate some of the potential problems with that. There are cases where it can be especially problematic, but that's the hope with this. And the umbilical cord is where it then enters into the embryo's body. So you can get things like nutrients, you can get oxygen, all of those things you need for growing a new human being. And during this stage, we start seeing that development of organs and systems. And if you delve even deeper into some of these stages, you'll start seeing things like vestigial traits. So we have a period where we have what looks like a tail, and that ends up being our tailbone at the base of our spines. Or even things like we have webbed fingers and toes for a period of time, and then we have a gene that tells our bodies to go in and have those cells all die off, so we have separated fingers. And even things like we'll have sort of gill-like structures on the sides of our necks that go away as we develop. So there's all sorts of weird things that occur in the background and then go away depending on timing. But yeah, during this stage we have two principles that we care a lot about, and this just talks about our tendency for development to occur in predictable ways. So two different terms, but the exact same logic, that's stating that we grow the structures that are most important to survival first, and then everything else can come later. So we prioritize things that are required to survive, and then everything else doesn't have the same pressure. So we can say that maybe there was a selective pressure where if, say you start by developing your fingers and toes first, if you were born early, you wouldn't survive. So any genes that encourage that pattern of development wouldn't get passed on. So we end up with our cephalocaudal principle of development, and this is just saying from our head down to our, the word is tail, but it's really to the bottoms of our bodies. So we develop our brains first, the important parts, and then everything else is going to be developed. The other way we can look at this is the proximodistal principle. And this goes from the proximate, so middle component, out to the distal parts of our bodies, to the outsides. So this is going to be, so our internal organs are going to develop before our fingers and toes. So once again, we prioritize those important to survival areas. With our prenatal development, though, we already got through 2 of our 3 stages. So we started with our germinal stage, where our organism is called the zygote. That's the first 2 weeks, and that goes up until implantation. Then we have the embryonic stage, where it's the embryo, and that's from our 2 weeks to 2 months. And then from that 2 months onward, we're in the fetal stage, where we call it a fetus. And this is that sort of last stage where we have our refinements up until birth. So during this stage, like we said, this is refinement. This is where we are adding some of those details. And we can see that we're seeing our muscles strengthen, our different organ systems are developing. We do see that our eyes start opening by around that 24-week mark. And again, I'm not sure if we've mentioned this before or not, but when we look at some of our senses, some of them have had some exposure, some of them have started functioning before birth. The big one that we talk about is our sense of hearing. where we can see familiarity with different voices and even with different songs and stories right after birth, telling us that before birth they have that recognition, they have that understanding and that exposure to different sounds. But with our eyesight, even though our eyes open and we start having some of the refinement of some of our visual systems, we're not actually making use of our eyes until after birth. We specifically need exposure to light and the stimuli that come with it in order to have proper development of our sight systems. So our eyes can open early, but aren't really used until after birth. And we'll find that there's a pretty rapid development of our vision. So right away we have very poor vision, but by the time we're, I think, 2 or 3 years of age, we're at like 90% of adult vision. So our refinement happens quite quickly. But I don't want to get too far ahead from that. When we look at viability, you might notice that some of the earlier documents have a different viability period. And that's because it depends on what they count viable as. So I think I have in my notes here, when they're talking about viability, I think this is sort of an 80% survival rate, where some of the earlier ones have a lower range. And so viability can mean different things depending on what percentages they want to talk about. So just in case you noticed that, I don't do too much with numbers, so I don't mind too much that we have different ones. I just figured I'd acknowledge that. One more thing I'd like to point out on this slide is that if we look at the brain development here, so we do see some of that refinement in the face, but with our brains, we start out with smooth brains, and those are then going to develop some of those bumps and grooves that we see with adult brains as time goes on. And if you think about it, if we have sort of premature babies able to survive after birth, at the time they might have fairly smooth brains, but that doesn't mean that they will always have smooth brains. Our development will progress pretty consistently after birth. So starting early, so being born prematurely, doesn't necessarily mean that you're never going to catch up to others of your age. It just means that there will be a little bit of a lag to begin with, but over time you end up kind of equalizing. And we'll actually see something very similar with our visual development curves, where children that are born with, say, cataracts that interfere with their eyesight, they do start with much worse vision for a longer time at the beginning, but after a couple of years, they've caught up and have equivalent vision to others of their own age once that development has occurred. So all of this ends up tying into that sort of plasticness of our abilities when we're quite young. And for us, plastic meaning flexible and changeable. So we have lots of plasticity within our brains at this age, and it allows us to adapt and change and continue developing even under different circumstances. I've just had in the past students point this out, that if that's viability and their brain is very smooth, what does it mean in the long run? Pretty much nothing, because they will catch up. So our next little chunk here is a very brief dabble into some genetic stuff, but specifically we're focusing on genetics and how it relates to sex determination. So I think we've mentioned before that we have our 46 chromosomes, and they exist in 23 pairs, or 22, the first 22 pairs of chromosomes. Those are going to be our non-sex chromosomes. Those are the ones that have most of our information for things other than sex. There are a couple of traits, a couple of genes that are found on our sex chromosomes, but the vast majority are on the others. Our last pair, that 23rd pair of chromosomes, here's where we have our sex determination. So XX or XY, XX being female and XY being male. And I'm sure nobody is curious, but because I have my bird thing to talk about, birds have WZs instead of X's and Y's. I have no idea why they named it that way, but their sex determination is opposite humans, where I believe males are WW and females are WZ. Again, oddities that you learn when you work with birds. But for us, with humans, it's XX or XY. And one of the really important things to note, and I suppose I can hop to this slide to show you. So this image here, this is called a karyotype. And what it means is that we've taken a single cell in a single nucleus when it's at that point when the cell is about to divide. Because during our normal life cycles, our cells, the DNA in our nucleus won't be as compacted and visible as it is in this situation. Obviously, this has been colorized for visibility. But during normal day-to-day stuff, our DNA is going to exist in a not so organized form. So we need to time it right to be able to see all of the chromosomes, and then you can spread them out and pair them up. So here, to avoid redundancy, we've shown one of each pair, but for any individual, we would have 2 of this and 2 of that, and so on and so forth. And these are the 22 of our chromosomes, and they're arranged in sort of the longest to shortest. And if you've taken sort of biology/genetics courses, you might know that they label these in terms of sort of their long arm versus their short arm. So you might see if we talk about a gene and a location, we'll say chromosome 7, and it would be either the P or the Q arm. And that just tells us above or below that centromere, that pink bit in the middle. And then each of these bands, like I said, artificially added, but the bands are kind of like a roadmap. So they tell us that in these bands, they are a known distance away from the center. So you could say gene on chromosome 7, this P arm, band 8. And then a certain amount of distance within that band, and that gives you a location to look for that specific gene. So this just helps us with organization. We typically won't talk about this at all. I think maybe once or twice we'll mention when we have a specific gene related to a specific trait that this is the location that it's at. But you know how I am with numbers and things like that. I don't really ask about them. But if you do see them, that's why those are mentioned the way that they are. But yeah, so these are our chromosomes. We won't care too much about them because we're looking at our sex determination, which are the X and Y chromosomes here. And unlike the other pairs where we're going to have matching pairs, unless there's something weird during crossing over and something goes wrong, all of the others will have equivalent pairs. They're going to look the same. It's the exact same chromosome, maybe with slightly different alleles, versions of their genes, but for the most part, same size and shape, except for the sex chromosomes. And so you'll notice that our Y chromosome is much smaller than the X chromosome. And because of this, they have very different contents. So while we might look at, say, a pair of chromosome 1s, and both of them would have a version of gene A, with X and Y, we might have a gene that's found on our X chromosome that's missing from our Y chromosome. Technically, you can have some on the Y chromosome that are missing from the X, but because the Y chromosome is so much smaller, that's a lot more rare. It's a lot less likely to occur. But we do have quite a few genes that are found on those X chromosomes that don't have an equivalent on their Y chromosome. And if you remember how genes work and the fact that we have 2 copies of each gene, we might have, say, a mutant version or a modified version of a gene that maybe doesn't function normally. So that's probably recessive. Most of them won't be expressed unless it's by itself or with something that also doesn't work. So it just means that one copy that works on the other chromosome means that we won't see any change in expression. So as long as we have one functional copy, for most of our traits, we're still going to have normal behavior or normal physiological expression or something like that. But if we have our X chromosome and there's no equivalent on the Y, all of a sudden a single copy that doesn't work is now going to show some change in some trait. And so colorblindness is one of the most common ones to talk about, where it is far more likely to see colorblindness in males than females. So a female can be a carrier for colorblindness and show no outward effects because they have 2 copies, and as long as one works, they are not colorblind. But if they have a son who inherits that, they inherit that defective version of that X chromosome, they will be colorblind regardless of what they get from their father. So you'll find that it's much more common, and it'll usually pass down through that male line. So we have a bunch of different sex-linked traits, sometimes also called X-linked traits. And like I said, there are some that are Y-linked, but they're very, very rare. But yeah, so that's a little bit of information about how these different chromosomes work. And when we look at those X's and Y's, you can see that in a son, you would have to get the Y chromosome from your father, because they would only be able to get a Y chromosome from their father. So their mother would give them one of the two possible X's. And then if you look at a female, then father passes an X and the mother passes an X to get XX at the end. All right, before I go too far off into genetics land, we can talk a little bit about how the Y chromosome can be very important. And that's because it contains a gene that controls this testosterone determining factor, or TDF. And this is really important because with our fetuses, they always start with the default of female, unless there is the presence of this testosterone determining factor that would activate by a certain threshold. So if there is no if there is no Y chromosome and there is no TDF present in that environment, then we would see development continue for female. Whereas if this is present and if that factor is then produced, then we see the development of male sex organs, so our primary sex characteristics. And once the testes develop, they secrete androgens, so male sex hormones, that are going to then have the rest of that development towards male physiology versus female. And like I said, there is a critical period where this needs to happen. So this TDF has to be present between the 6 to 8 week mark. And if it isn't present, or if there isn't sufficient levels of it present, then we have that default female physiology at the end. So there is that one narrow window where this can occur, and it specifically requires a Y chromosome that has a functional gene to produce this particular factor. So very precise timing for this. We can also have a couple of oddities when it comes to our sex chromosomes. You may have heard of different genetic disorders where you can have extra or too few copies of different chromosomes. For the most part, these are probably better tolerated within our sex chromosomes. With other chromosomes, others of those pairs, we really don't tolerate duplication very well. And different species have different tolerances for those duplications. If we look at, say, plants, plants have the ability to have lots and lots of duplications with no problems. But with humans, because most of our traits and our expressions are relying on having two copies, two different versions of a particular gene, having more copies than that can cause problems. So for the most part, when we talk about these extras or too fews in terms of chromosomes, it's mainly going to be for these sex chromosomes. So the first of these is the Turner syndrome, which is where we only have a single sex chromosome and it is only an X. So X on its own, no Y, no paired X. And so for this, you have an individual who's only having whatever's contained on that X. And if we don't have a Y chromosome present, we don't have that testosterone determining factor, and therefore we would have expression, physiology of a female. So there are other side effects that can come with this. You don't need to know tons of details about this, but they often have visible symptoms. Things like their ears tend to be a little bit lower. They are more likely to have slight webbing between their digits, and their hairline, instead of having sort of a clear distinction between their neck and the back of their head, they tend to be a little bit more close together. because these individuals don't have two copies of the X chromosome, while they will have that female physiology, they won't necessarily reach sexual maturity in order to be able to have their own offspring. So they'll usually figure that out as they get a little bit older and don't end up developing secondary sex characteristics during puberty. For our others, so that's the one where we have missing chromosomes. With extra chromosomes, there are 3 that are fairly common. The first being triple X syndrome or trisomy X, which just means you have 3 copies of an X chromosome. Once again, there's no Y present, which means no TDF, so we would have female physiology. This one is actually fairly common and occurs in 1 out of every 1,000 females. It's one of those disorders where it can go undiagnosed until somebody happens to look into your genetics and maybe checks for something like this. But it tends to have fairly mild symptoms that don't necessarily get noticed. Our next one is Klinefelter syndrome, and this is where you have 2 X's and a Y. And so once again, we have extra sex chromosomes. And here, having the presence of a Y means that you do get the expression of testosterone determining factor, so male physiology. However, it does also come up with some symptoms. And there are some odd ones here where they end up having weaker muscles, but also taller in terms of stature. So they might have trouble with movement because of that. They also have, again, sort of issues once you hit puberty and start seeing or not seeing the development of those secondary sex characteristics. So they might not go through puberty the same as you would if you only had 2 copies of your chromosomes or sex chromosomes. And then the last one being Jacob's syndrome, which is XYY. So here we get the extra Y chromosome. And this one, again, taller than average, but it tends to also come with stuff that's due to excess androgens, so the male sex hormones. So you can have things like not just taller height, but also more prone to acne, possibly due to higher levels of sweating. And so some of these aren't necessarily super noticeable. This one, there is no huge effect on fertility, unlike some of the others. So again, it can go pretty much undetected unless somebody thinks to check. And like I said, I've added sort of extra details. I went through on Wikipedia and pulled out some of the interesting side effects and things just for some flavor. But mostly we care about the fact that you can have more or fewer, and it can have some side effects, but not too much detail there. There we go. So next we can get into teratogens. So this is a whole bunch of different ways that the intrauterine environment can affect development. So teratogens are specifically things from our environment that can negatively impact prenatal development. And in these cases, there's all sorts of things that can count as a teratogen. There are some odd ones, things like malnutrition. While a lack of nutrients isn't necessarily some substance that's causing problems, it is a lack of substances that causes problems, so it tends to get lumped in here as well. I have some slides. I don't want to get too far ahead of myself just chatting about them, but one of the things to keep in mind is that with each of these different teratogens, there are variations in terms of the effects that they can have. So the dose, so how much is it that you're exposed to, the timing of those doses, especially very, very early on in development, so soon after conception, you tend to be very susceptible to the effects of teratogens. We can also look at cumulative effects. So a single dose might not be as harmful as long-term doses. And all of these different factors can interact depending on which substance it is that we're looking at. So some can be more harmful than others. But we will stress that with these teratogens, they're actually very difficult to study because you can't manipulate something like this. We are left with correlational studies where we would look at, after the fact, someone was born with some condition or some kind of developmental problem or change or whatever it is that we're looking at. And can we trace that back to some exposure from the mother during their pregnancy? And this becomes problematic because sometimes people will have exposures even before they know that they're pregnant. So all of those things make it very, very difficult to study these. So we'll keep that in mind when we look at some of the vagueness of our discussions in terms of things like dose and timing. But yeah, so lots and lots of correlations. For some of our specific examples, drug use, and this can be prescription drugs or non-prescription drugs. So this could be things like recreational drugs. It could even be alcohol. And alcohol is one of the ones that's very widely studied simply because it's a pretty widely used substance. And like I said, where a lot of people might not know that they're pregnant at the very beginning when we would have an increased susceptibility to the presence of alcohol, you can have exposures causing problems. You may have heard that there have been studies that say some moderate alcohol consumption after a certain stage is safe, with air quotes, and that's because we can't manipulate anything, and so nobody wants to say that there is a certain amount that is safe or a certain time period that is safe. It's always better to sort of err on that side of caution, which is why they widely encourage people to avoid any kind of drug, including alcohol, any time during pregnancy. This is also one of the reasons why, if people are trying to get pregnant, they're told to avoid these substances, because not only can it negatively impact the possibilities of getting pregnant, but there is that potential for massive effects early on. So all of these play a role. There will be probably a lot of studies coming out in the coming years to look at things like how marijuana will affect pregnancy, especially with recent legalization, because it would have been something that wasn't as common before that, and now they want to see, will there be effects? And again, correlational studies here. So that was drug use. Actually, do I also have, okay. I do have it on the next slide, but it is technically a drug. So thalidomide is one of those sort of case studies that we talk about in terms of teratogens and how significant of an effect they can have. If you've never heard of thalidomide, this is a drug that was prescribed back in the 1960s, and it was actually very, very effective at reducing the effects of morning sickness. One of the problems with this drug, though, is it's a single chemical molecule, but if you've ever taken a chemistry course, you might know of the concept of chirality, and that just means that you can have mirrored versions of a particular drug or substance of some kind. So this molecule can exist sort of in one form, or it can exist as its mirrored opposite. And if that molecule isn't symmetrical, the two have very different shapes because they're mirrored. And in this case with thalidomide, one shape, one half, ends up being very effective at treating morning sickness, and the other shape, the other version, ends up causing really severe birth defects, and especially those that end up affecting the development of limbs. So what they figured out is, after this was approved and put on the market, is once they weren't so cautious with which version was being put out, they had massive amounts of these birth defects. And it ended up being sort of a huge lawsuit. It affected thousands and thousands of people, mainly in the US, and they sort of had to pull this off the market very, very quickly. But it had this massive effect on development, and so it showed us that different substances can have a very detrimental effect. So yeah, that's one specific case of drug use that can have that effect, and different drugs may have different effects. We can also have maternal exposure to illness and toxins. There are some really neat studies that look at correlations between different types of illness and different kinds of outcomes later on in life. I recently saw a study that looks at influenza rates. So if they correlate or make an analysis of the strains and the intensity of influenza in the population, and then rates of, I can't remember if it was depression or schizophrenia, but the two end up correlating, where if there are more cases of influenza and if the strain is an especially aggressive strain, there tends to be an uptick in certain psychological disorders sort of in the future. So if you can trace back to parents who might have been, or mothers who would have been exposed to this flu, it tends to have long-term effects on their children. What else? Oh, and so looking at sort of other things, including STIs, different illnesses that a mother might have could either be passed on to their children or end up affecting the environment inside their bodies that would then affect development of the child indirectly. Both cases can have an impact, and then different illnesses will have different effects depending on sort of severity and timing and all of those other things we mentioned. We did also mention that malnutrition is one. Malnutrition is actually one of the most impactful effects on development for pregnancies. And the reason for this is because when we have all of those different stages of pregnancy, you have very, very rapid cell division in a very short period of time. And so we're making lots and lots of copies of these cells, and each copy is taking up a lot of resources. And so if the mother doesn't have the resources to provide, then you can have a lack of development. You might not be able to have the cells divide the way that they're supposed to. Or you might have the cells there, but you're not able to make the proteins that you need to keep them running optimally, or whatever it is. So depending on what you might be deficient in, you can end up with different effects on development. And speaking of sort of lacking different nutrients, some people have linked those sort of deficiencies with things like cravings during pregnancy. Where if you get into cravings, there's actually a learned component. So even outside of pregnancy, if you are, say, iron deficient, your body might have learned that you can get iron from eating green leafy vegetables or red meat. And so when you are iron deficient, your body craves those things. So it's a combination of classical and operant conditioning to get you the nutrients that you need. And the same logic has been applied to pregnancy cravings, where you would crave these things that would give you whatever nutrients it is that you're lacking. So you might get those odd cravings for things that you would never have had before, or in the same quantities or combinations. But if you are deficient in certain nutrients that you only get from those foods, that could explain why you have such a strong inclination towards them. That doesn't always explain everything, because some people will have cravings for stuff that they've never tried before. So there's maybe sort of an environmental component or a cognitive component. But yeah, there's a couple of different studies that look at stuff like that. What else? Oh, so even with the malnutrition, before I got off on my tangent, malnutrition is not just sort of showing sort of lack of development. It can also have an increased risk of birth complications, sort of long-term effects. It can also be related to rates of miscarriage as well, which pretty much all of the teratogens can be tied to, depending on, again, which ones and dosages and stuff like that. So lots and lots of potential to affect a bunch of different stages or aspects of pregnancy. And then back to this slide, where the last point being the Zika virus. This is one that had fairly recent interest because there was an uptick in rates of this virus being spread. And then we started seeing that it had very large effects on birth, specifically microcephaly, which just means that while the spinal cord develops, the brain doesn't end up developing the way that it's supposed to. And so a child could be born with sort of a almost caved in looking skull because the brain isn't there to fill that space. And again, variations in severity depending on sort of the infection and how long it's been present and things like that. But it was definitely something that was very well studied because it became very predominant very quickly. For other things, just in my notes here, so I don't know if I explicitly stated it, but when we were talking about drugs, nicotine can also be considered in that category, because nicotine is a drug. So smoking is linked to higher rates of miscarriage, higher likelihood of premature birth, low birth weight, and higher likelihood of cleft palate, as some of the more obvious ones. You can also have things like exposure to heavy metals can also be problematic, because again, they can get incorporated as cells are dividing, as things like bones and muscles are forming. If it's around, it might get incorporated and cause problems. And this can be especially problematic in areas where maybe you have a lot of metals in the soil or in the water. I know a little bit about this because before I got back to school, I worked with the federal government and we did lighthouse remediations, which is their fancy way of saying that we investigated whatever ended up in the soil around lighthouses in Nova Scotia. And there was a lot of mercury around because, if you didn't know, lighthouses, when they have their big spinning lights, they used to use mercury as a lubricant. So they'd have a pool of mercury that the light would spin in. And when they were done, whenever it was mucked up with whatever had broken off the light, they would just chuck it out the window and replace it with new stuff. So clearly that would cause problems in the soil. But we also had very high background levels of things like lead that ended up causing problems with water sources, because a lot of people use well water. So you'd have to be really cautious of things like that for those who were pregnant or trying to get pregnant, because you end up with high levels of heavy metals, and it can be in your tap water, which is, again, problematic. So things like that you might not consider problematic, but can absolutely be there in the background. Radiation is another one that we didn't explicitly mention, but would obviously have an effect. So exposure to radiation can be problematic. It's one of the reasons why people are very reluctant to give pregnant women x-rays, because while the radiation level might be acceptable for an adult, small amounts of radiation can have really large impacts on growing and developing fetuses. So all sorts of things to consider, and a lot of it ties back to that idea of very rapid cell growth, rapid division, and high pull on resources. So even small amounts of things can have large impacts just because of that sort of condensed occurrence of all of that development. All right, so no more tangents on that topic. Now we can continue from the point of birth forward, so infancy and then into childhood. And so here we have this very rapid development following birth, where, as we said before, we have our brains are starting to have those different bumps and grooves, and we're going to start seeing lots and lots of connectivity between all of the neurons that are in our brains. And if you keep in mind, I guess if you've encountered infants, they are always very cautious of an infant's skull, because they're sort of the plates that become their skull haven't quite fused yet. And that's partially because you need that ability to collapse a little bit in order to give birth properly, but also our brains end up growing very quickly after birth, to the point that it's not so much that we do see a little bit of a growth in the number of neurons, but we also see that the cells themselves get much larger. So we have lots of tiny cells because they grew very quickly and didn't have time to build up all of their cytoplasm, all of the fluids inside. But as the growth rate slows down, now they can get larger and pull in the resources that they need. So we need that adaptability as our brains reach sort of their full sizes. So there's a lot of different changes in terms of that. So not just connectivity, not just number of cells, and not just size, all three of them interacting together. So this process means that we have very, very quick changes in a lot of different areas very quickly after birth. And so we're going to start talking about cognitive development and also getting into the perception changes that come along with this. So our definition for cognitive development is just that we're looking at how the brain develops and how the different ways that we think and perceive our environment and react to that environment are going to grow and change as we get mature. So we're going to look at a little bit like our information processing. We'll look at some problem solving, some language learning, which means that this overlaps a lot with some of the stuff we talked about in earlier chapters, which is really nice because we get to sort of acknowledge those sections and move on to more interesting topics, or at least different topics that are interesting because they're new. But if we start at the beginning with our perspectives on newborn sensation, back in the late 1800s, William James was one of our fairly vocal individuals talking about perception, and he suggested that when newborns are born, they are surrounded by stimulation that they have no idea what to do with. So the specific quote is, they are surrounded by buzzing, blooming confusion. So they're hearing sounds for the first time and they don't know what to do with them. They're seeing lights for the first time and it doesn't mean anything. All of these different sensations have no context and therefore have no meaning until they've experienced things, until they've interacted with things, until they've learned what each of those sensations mean. So this is framing newborns as passive, and they have disorganized perceptions, and that they would start at the beginning with no experience, nothing to frame all of that incoming sensation. So this ties nicely to that idea of tabula rasa, starting with a blank slate, where you would have to acquire all of your associations through learning and experience with your environment. Though, of course, at this point, we've talked about enough things that seem to have innate components that we wouldn't necessarily believe this is the whole case. So instead, we've mentioned the fact that we have auditory stimulation before birth. And we know this because we have seen recognition and familiarity with certain stimuli, sound stimuli, after birth. We also have some research that's looked at things like tactile stimulation and chemical stimulation, where we can have, right after birth, you can look at 1 or 2 day old infants who will react differently to sweet things versus sour things. So they can tell the difference and they respond positively to sweet things and negatively to sour things right off the bat with theoretically no prior exposure. And with tactile components, you'll see things like some thumb sucking, which I always say backwards. Thumb sucking is something that happens even before birth and can continue right afterwards, and things like that, or grabbing your feet, those are all tactile stimulations. And we'll see that some of those actions, we'll end up calling them, I think they're primary reflexes or something similar. Those tend to occur fairly quickly and they're very repetitive. So it tells us that there's something there very early on. So we know that some of our systems are stimulated within the womb and can be operational at birth. And I did mention that with our vision, it isn't necessarily operational at birth, but it is definitely starting. And when we look at, say, neural development, we start to have some of those connections forming within our visual system even before we're exposed to light on a regular basis. So that process is there, the groundwork is laid, we just need that exposure right after birth. So this idea that we're born with nothing and we start from scratch is definitely not the case. So since we've touched on the visual system, we can actually go into some of the details here. So our visual system, we start out with 2800 vision. And so if you think about it, a standard adult should have, theoretically, 20/20 vision. And that's what, say, glasses are going to correct your vision to. And what that just means is that at 20 feet away, something looks like it's 20 feet away. You're capable of seeing it as it should be seen. So if we have 2,800, that means that at 20 feet away, it looks like it's 800 feet away. So it is a very blurry and imprecise and non-resolved mess. And again, that kind of makes sense at birth because they've had no exposure, no stimulation, no ability to use that particular sense yet. By the time they're 6 months old, we're up to 2,100, which is a pretty large improvement in such a short period of time. One thing to note, that right after birth, and especially in those first few months, we do have a sort of sweet spot where our vision is most sensitive and most accurate. And that distance ends up being about 20 to 40 centimeters from the front of their faces, which, if you work it out, ends up being about the exact distance between the baby and their mother's face while breastfeeding. So we could see that maybe there was a selective pressure that encouraged them to be able to see and start recognizing their parental figures early on, especially if sort of connecting to them and being familiar with them is something that would help the infant survive. There's also sort of evolutionary tales about different traits of infants, things like the fact that they have really large eyes or the proportions of their faces that not only sort of help them recognize their parents, but make their parents see them and want to protect them. We have sort of a back and forth selective pressure there that encourages that parent-infant bond. But sticking with our vision discussion, this development from very, very poor vision at birth to still poor but better vision a few months later, this is going to end up continuing or developing in a continuous fashion. So we see this gradual increase. The more they're exposed to things, the better their perception gets, the more stuff develops, and so everything gets more and more accurate in a pretty progressive way. We do start seeing some of their abilities to not just see stimuli, but also to start interpreting and making connections between stimuli fairly early on. So you might remember Gestalt principles of organization. And these are rules about how humans have a tendency to group items together following certain principles. So things like the principle of similarity, where we would recognize a whole bunch of the same thing as being belonging to a group together. So if you looked at a bunch of blocks, you might see that all the square ones belong to a group and maybe all the triangle ones belong to a different group. So we start seeing that sort of pattern recognition within 3 to 4 months. We can also see contours and also the idea of closure, where we assume that certain shapes will continue even if we can't see the whole shape. So if you see a almost closed circle, our minds will interpret that as a full circle, even if we are missing that last little piece to close the circle off. And the logic behind it is that in our environments, we aren't guaranteed to see the entirety of any given shape. There's the possibility that it could be obscured by something else in the environment. So it makes sense for our visual system to fill in some blanks. And our brains are very good at filling in blanks. So things like closure or finding those contours, even if there isn't a clear outline, it makes sense that that's something we'd be predisposed towards, especially where we see consistency across cultures for those types of principles of organization. It tells us that there's maybe some biological component which could help explain why we develop that ability so early on in life. Possibly some kind of biological framework, to use some of the terminology we've used before. We also start seeing very, very early on a preference for faces or face-like stimuli. Once again, the earlier that these things develop, the more we assume that there's going to be some genetic or biological component here. So within the first few months, we see that they're going to preferentially view faces. So if they can choose between looking at a shape that has sort of 2 eyes and a nose and a mouth in a face orientation versus even if they have the exact same features but rearranged so it's not like a face, they will look at the one that matches face proportions for much, much longer. So they show, using that looking time paradigm, a preference for face features. And that starts very early on, and it develops. It becomes even stronger as they get older, to the point that once they have sort of the visual acuity to perceive more detailed faces, they end up showing a preference for detailed faces over simple faces, but they still prefer even the simple faces over sort of more boring stimuli. Or again, even if we're controlling for complexity, the same face pieces, so a cartoon set of eyes, nose, and mouth, if it's not arranged like a face, it is far less interesting to them. So something about that face orientation makes it important. And we shouldn't be too surprised, because I typically talk about our fusiform face area. We have part of our brain that lights up when we are exposed to face stimuli. And that shows that we have some kind of connection, some kind of importance put on those faces. There is a caveat that that area might be due to experience rather than just face specific, because you don't need to know this right now. We might have mentioned it or might mention it soon. But if you train someone up to recognize different types of stimuli, you can get activation in that area as soon as they become an expert at looking really closely at that stimulus. So we could frame it that we have activity there because we see faces all the time and are experts at recognizing faces. They're not quite sure if it's sort of that the area causes the face specialization or if seeing lots of faces makes that area activate. Both could be possible. But yeah, that comes up a couple of times because it's one of those specialized areas of the brain. But yeah, so we will have, I think there's a figure in just a few slides where we can actually see those different preferences using that looking time idea. And I think we mentioned this before the break, where we had some of our different standards for measuring things like this, where our preferential looking procedure or a looking time paradigm, we just say, showing you different stimuli, what do you look at for longer? You can also do it with, say, simultaneous presentation. So if I show you two things, which one do you stare at instead of the other? That can be used as well. But all of them are basically focusing on that idea that infants will look at things that are more complex, or they will look at things that are familiar to them. So we can use that to do some testing. And one of the things that they can use that for is actually determining how much detail an infant can see. So if this diagram here is showing what we have figured out that an infant can see for the first one at 1 month of age, so that's this one is 3 months, and this one is at a year of age. And the way they can tell this is, say you have a 1-month-old and you're going to show them this image that's really, really blurry, and you're going to show them this image that is really, really sharp. If their visual acuity, if their vision is not very detailed, if they can't see things clearly, then both of these would look effectively the same because they don't have the ability to see these extra details, so they would look at this image and this image for about the same amount of time. Whereas if they could see about this level of detail, showing them this versus this means that the blurrier image would be less interesting. There's less detail to see. They could see a little bit more detail, about this amount of detail, and therefore they'd stare at the clearer image for longer. So you can have series of more or less blurred images and ask, by showing and measuring their looking time, which one is most interesting. And that would then tell you, are they capable of seeing this level of detail or not, through all of these different comparisons. So once again, we run into that issue of we can't ask that direct question and say, can you see this or not? But we can use their behavioral response to tell us, can they tell the difference or not? And if they can't tell the difference, then they're not capable of perceiving that level of detail to be able to make that discrimination. So that's how we can use this to determine when that detail becomes interesting, whether or not they can see that level of detail. And then this is that other diagram I was mentioning, where we can look at, again, preferential looking. So if we show them a cartoon face, how long do they look? A very long time. We show them something complicated. So here, a bunch of sort of squiggly lines, they'll look at that for quite a while. Bullseye is very interesting as well. But if we show them something simple like just different colors, they look at those for a much shorter period of time. Though we can find that different colors might be more attention-grabbing than others. And once again, this shouldn't be super surprising because in a lot of species, red ends up being a very attention-grabbing color. And so they would stare at red for longer than, say, white. And so with these tasks, you can show them a whole bunch of different stimuli, whether it's one at a time or choose between the two, and you can get that preference for which one is more interesting, which one do they stare at for longer, therefore that is somehow important or fascinating to them. So a lot of our determinations of what they are capable of seeing comes from tests like this. So yeah, we get all sorts of things like this. And like I said, so we start off with cartoons, and eventually, as their vision gets better and better, you could show them more realistic faces, and those are then faces and more detailed, and would then have sort of an even longer bar once they're capable of seeing that level of detail.